Hey there guys, my name is Tom. You might have found my YouTube videos through my TikTok or my Instagram. If so, thank you very much. So this is the very first video I'm making on a series of videos for crash coursing the whole of the GCSE, AS and A2. This one is cell structure for AS, and there will actually be a small second part to this video because we will have a practical skills on microscopes to look at as well. But this video will cover all of the content for cell structure, as you can see in the title up here. So let's make a start. So I've moved myself down to the bottom of the screen here so you can see what's on the, I suppose, your board a little bit better, or your YouTube screen. And as you can see, and as per this subject title, we're looking at cells, which means we need to understand the structures that you find in animal cells, plant cells, and bacteria, which we're going to look at being called prokaryotic and eukaryotic cells a little bit later. We're going to compare all three of those, as well as viruses and their structure, although we won't call viruses cells for now. And then we'll look a little bit at the way that the microscopes work. However, it's not specifically the skills that you need for microscopes. We're going to cover that a little bit later in a practical video, which hopefully will help you too. So let's make a start. And so your objectives: know the names and functions of all of your organelles. That's going to make up most of what we need to learn straight off at the start of the unit. We're going to compare all of those organelles. We're going to add in evaluating viruses as a living organism based on the structures that they have and how they live, or how they exist, I should say. And then last but not least, as you can see, describe the differences between microscopes, and then look at magnification and resolution as a learning point rather than as a skill. As we're working through the video, don't forget, if anything is bold, if anything is red, or if anything is in italics, it's probably related to how you need to use it in the exam, or that it's important for learning. So just pay attention to that little bit on screen there, and make sure that you do take notes on those bits as we're going through. So we're going to take a look now at our first section of the lesson, which is on all of the organelles that can be found in both animals and plants. I'm just going to disappear for a second so that you can see that list there, and we will be working from this diagram. Now, just to make sure that you're not confused, it is an animal cell, but these organelles can be found in both animals and plants. So when you're making your notes, make sure that's really clear, guys. So I've just made my little circle around my head a little bit smaller so that you can see the diagram above. And if there are any other diagrams whilst we're going through organelles that you can't see, I will disappear a second so that you can make drawings and notes. You can see that we're looking at mitochondria, and our function remains very similar to what you learned at IGCSE or GCSE, in that it's the site of aerobic respiration. A slight little bit of extra information: you can continue that statement to say that the aerobic respiration, or the mitochondria that carry that out, convert substrates such as sugars into ATP. And I want you to make a big bold note of ATP, because you're replacing the word energy with ATP. Energy isn't just some magical thing that exists in your body. It is a molecule. That molecule is ATP. We will look at its structure in a little bit more detail later on. You can also see I've made a note there about the photomicrographs. This is the sort of photomicrograph you'd be expected to interpret as well as recognize in a cell. And you can see that the mitochondria has that very distinctive double membrane, which you'll be expected to notice. And you can always tell it's the mitochondria because it's got those big folds in between those membranes. Next up, the cell membrane. Again, an organelle that you probably remember. The cell membrane's function is to control the movement of substances into and out of the cell. We usually call the cell membrane permeable, and in certain circumstances it's semi-permeable, which means things can move in and out. The photomicrograph is usually very easy because it is just that layer around the outside of the cell. One thing to note here is that it sometimes, under very powerful microscopes, will appear as a double line. We will look at why it's a double line in the next couple of units. But for now, nice and simple, just remember cell membrane is around the outside of those cells. Another similar to IGCSE organelle that we have is the nucleus. The nucleus still contains the DNA and it still controls the activity of the cell. However, there are a few parts to the nucleus that we now need to be able to identify. The first of those is the nucleolus, which you find as like almost like a nucleus inside of the nucleus because it's a big black area, and its job is to make ribosomes. You should know ribosomes from IGCSE, but ribosomes we will look at as a separate organelle in a moment. They are made here in the nucleolus. The membrane that goes around the outside of the nucleus contains nuclear pores to allow things such as sections of DNA called mRNA and those ribosomes to move in and move out of the nucleus as and when they need to. And so you can see all of those sections quite clearly on this micrograph picture. You've got a nuclear membrane around the outside, and whilst it's not perfectly clear, there are areas that are not whole. The membrane has been split to allow things to leave, and that big dark patch that you can see slightly to the right of the center is the nucleolus, which is what's going to carry out all of that ribosome synthesis. To help you and to make sure that you've got all of that nailed down, here's a quick clarification point. This is the structure of the nucleus that you need to know it as, with chromatin or DNA, the nucleolus in the center, and a nuclear membrane with nuclear pores outside. And if you wanted to make a note, here are your structures that must be remembered, okay? The function is on the previous slide, but this is an important one because it does come up often and it's quite different to our IGCSE. Our next organelle is the rough endoplasmic reticulum, or RER. Now, we state that the RER is the site of protein synthesis. However, in a moment, we're going to take a look at ribosomes. Ribosomes are the actual organelle or structure that carries out the protein synthesis. The RER is just where that happens. The RER is like a big membrane, and the proteins get made into the membrane, and then they can be transported elsewhere for being put together into different other things or bigger proteins by one of the organelles we're going to look at in a minute. You can see that those ribosomes are really clear on the photomicrograph as black dots around the outside of a membrane structure, and that structure usually exists quite close to the nucleus if you're ever looking at a whole cell diagram. So we just looked at the rough endoplasmic reticulum. To confuse you, there is a smooth endoplasmic reticulum as well, abbreviated as SER. The rough endoplasmic reticulum, we just mentioned, synthesizes proteins. The smooth endoplasmic reticulum synthesizes lipids. That's the basic thing that you need to remember, is synthesis of lipids. However, there are some other functions that can be mentioned, such as detoxifying the cell. Basically, that means is if the cell is making anything through metabolism which is harmful, it can be gotten rid of or broken down in the smooth endoplasmic reticulum. For the photomicrograph visual, it sits a little bit further away from the nucleus, but most often it looks very, very similar to that membrane-bound ER that we were looking at before, but there's no ribosomes here this time, so there's no small black dots. That's usually your big giveaway for this one, all right, guys? And another membrane-bound organelle is the Golgi apparatus, sometimes called by Cambridge the Golgi body. However, you can always say Golgi apparatus, you'll definitely get the marks. Its function is to take all of those things made by the endoplasmic reticulums, both the smooth and the rough, modify and package them, ready to send them somewhere else in the cell or outside of the cell if it's secreting. And so modify, sorts and packages is good wording to remember, and that's why I've added that in bold for you. For the photomicrograph visual, I'm going to disappear for a second. This is very similar to the SER, except that it's going to be a little bit further away from the nucleus on a diagram, and its shape should represent a Wi-Fi symbol. It's got those big curved areas at first, and then those turn into smaller vesicles which are coming away from the Golgi apparatus. And just to signify that, I've added that little cartoon of a Wi-Fi symbol there to help you remember. Hopefully that helps. We've stuck with the same symbol here. Those little bubbles or vesicles that come away from the Golgi apparatus, usually they are vesicles containing products. If they contain a digestive enzyme, you should all remember what an enzyme is, then we have a different name for them, and that is a lysosome. That's because they do a specific function, and you will need to refer to that later in AS, particularly in the immunity topic, when we're looking at the function of phagocytosis. But yes, lysosomes are coming away from the Golgi body. So I've used the same diagram there for you, and you can see those little buds that come away are what we call lysosomes, if they contain enzymes. Now that we're finished with our membrane-bound organelles, we're going to go back and jump to ribosomes. Ribosomes are the organelle that carry out the protein synthesis. Now, you can see in my notes here that I've actually written two large numbers, 80S and 70S. 80S are the size of ribosome which you will find in eukaryotic cells. Later, we will mention something called a 70S. It carries out exactly the same function, which is protein synthesis, but it's found in prokaryotic cells. We will come to the bit about mitochondria and chloroplasts later. Photomicrograph visual, I've used exactly the same one that you will recognize before. They are the small black dots which you find on the outside of the membrane of the rough endoplasmic reticulum. Okay, and now we're going to move to some very strange new organelles that you almost certainly haven't heard of before. The first of those is the centrosome. Now, it's a bit difficult with the wording here, and so I'm going to clarify that for you afterwards, but the function of the centrosome is to make spindles. Spindles can be pathways in the cell, which vesicles, lysosomes can follow for transporting different things around the cell, and it can also be related to mitosis. That's topic 5 in AS. Mitosis being the process that pulls the chromosomes apart. Now, the spindles are the things that can pull the chromosomes apart. We'll look at that in more detail later, but that's the function of the centrosome. Interestingly, for the photomicrograph visual, you need to remember a set of numbers. That set of numbers is 9 times 3 for each of the centrioles. Now, what do I mean by centriole? I've just said there. You have got the centrosome, which is the organelle region, but the centriole is each of the 2 sets of cylinders or spindle fibers that you can see above. Now, each of those spindles or microtubules is called a spindle or a microtubule. So this is just a quick clarification slide for your notes. Remember that you have got 3 terms here. The organelle, which is the region containing 2 centrioles, that's the centrosome, and each centriole is made up of 9 sets of 3 microtubules. Hopefully that makes sense, guys. If you need any clarification, please drop me a comment. And we've got 2 more just to check for the organelles for eukaryotic cells. Cilia, you've probably heard of these before. Cilia are little arms which come away from the surface of the cell membrane, and they move substances around. So their job is basically providing locomotion by pushing other things. And photomicrograph visual, I've got a whole cell diagram here for you. This is actually a diagram of your respiratory airway or your trachea, and you've got lots of cilia attached to the top of those cells there for brushing away mucus. Last but not least, we're going to look at the microvilli. They are found on the villi which are in the intestines. Now, microvilli are very different to cilia but can easily be recognized as one another because they look so similar. Villi are simply folds in the surface membrane of a cell which increase its surface area. Usually this is for absorption, hence why we find them in the intestines. It is worthwhile mentioning, as I've said, I've written there villi brackets micro. The main structure that you can see is a villi, with each cell on the villi having a microvilli, which are the additional folds in that surface membrane. Make sure you can understand the difference between the two. And our photomicrograph visual just here, I'm going to disappear for a second for you so you can see that clearly. Down the bottom in the left hand side you can see those distinguished microvilli folds. And so just to clarify exactly what I'm talking about, we've got 2 structures there which can extend from the surface of the cell. The cilia are for motility or for pushing things, and they're made of actually microtubules, which you should already know what they are. And then microvilli are just an extension of the surface membrane, which increases surface area for things like absorption. Make sure you make a note and know the difference between the two. Now, I did say we were finished with organelles, and that's because I want you to make a very clear note. The last thing we're going to look at is cytoplasm. Cytoplasm is technically not an organelle because it is just the fluid that is found within the cell membrane. And it contains lots of different organelles that we've looked at. It also has lots of reactions taking place, but independently, we wouldn't want to call this an organelle. So it's worthwhile remembering that you do have cytoplasm, but not necessarily calling it one of the organelles, particularly if you are asked in an exam question, because it's not, right? So, looking at this diagram again, you should now be happy with what all of those organelles' functions are and what they look like under a photomicrograph visual. That's for the shared organelles for animals and plants. However, as you may have guessed, we also need to know that the plants also have some specialist organelles. Those specialist organelles are listed here with a lovely diagram again for you, and we're going to take a look at these four in order: chloroplast, cell wall, plasmodesmata, and then our vacuole. So let's make a start with that. I just disappeared for a moment there to make sure you could read that list of objectives, guys, but our next organelle is the chloroplast. Again, you probably remember this one from IGCSE. The chloroplast function remains much the same in that it carries out photosynthesis for plant cells. It does contain its own DNA and its own ribosomes. We'll look at a little bit more about explaining that later, but for your notes, and you can put it in bold if you would like to, circular DNA and 70S ribosomes are found within the chloroplast. The photomicrograph visual, they look something like this. Similar to the mitochondria, they have a double membrane. If your diagram or your micrograph picture is colored, you will almost always see chloroplasts as green, and those chloroplasts have large, darker stacks within inside them, which are basically the sites of photosynthesis. That's where all of that green pigment is found. But it's usually quite an easy pickup to find, so hopefully you can recognize that when you look at a few more pictures of cells in the future. Cell wall. Another one that you probably do remember from IGCSE. Its function is to provide structural support to the plant cell. It's stopping it from collapsing underneath all of that water pressure, which is going to be pushing from both sides. I have included quite a strange looking diagram. You might be a little bit confused. This is actually the structure or the biochemical composition of cellulose. You do need to remember, and you do always need to refer to the fact that the cell wall in plants particularly is made of cellulose. So in red, and that should go into your notes as well, cell wall is made of cellulose in plants. Photomicrograph visual, usually quite an easy one to see. You've got that first layer around the outside, which is a cell membrane, because don't forget plants do still have a cell membrane, but around the outside of that and in between each of what looks like a cell structure is a cell wall, and that looks like that slightly yellowed block around the outside of each of those cells there. A very new term, plasmodesmata. Plasmodesmata are channels which exist in between all of the cells that pass through the cell wall. Plant cells, like our cells, animal cells, and other kingdoms, need to be able to communicate with each other as well as transport materials. But because they've got that cell wall in the way, it's quite awkward to send things. So instead they have channels which allow transport and communication. Couldn't find a great diagram for you, but if you zoom in a little bit on this diagram, in between all of those plant cells that you can see there are very faint yellow lines which are identified as plasmodesmata. And then last but not least, we have our vacuole. Again, you probably remember this run from IGCSE. The vacuole's job is to store cell sap. So that's nutrients, water, different products and waste products. But its actual function is to maintain cell turgor. Now, what we mean by maintaining cell turgor is kind of pushing the cell outwards onto those cell walls to make sure that the cells and the plant overall hold their shape and structure. However, please don't forget that you do now need to refer to that as turgor pressure or maintaining cell turgor, because that is scientifically the correct way to talk about it. You'll notice here that I've also written tonoplast. Around the outside of the vacuole, it has its own specialized membrane called the tonoplast, which basically controls what moves in and what moves out of that vacuole. Photomicrograph visual is usually very, very easy because it's a very large part of a plant cell. They occupy quite a big space. On this one, they've been stained pinky purple for you, so you can see it quite clearly. Well done, you've finished all of the animal and plant cell organelles. Great job, guys. Now is probably a good time for you to take a bit of a break, and as well as that, you probably want to review what you've learned before you move on to the next section. For that review, what I suggest you do, just going to disappear for a second, is label, define, and compare animal and plant cell organelles. So what you really want to be doing is finding some diagrams of animals and plants, particularly looking at both micrographs, so microscope pictures, and diagrams on the internet without labels, and seeing if you can add labels, as well as comparing animals to plants. If you are working along online on my course, the worksheet will have a task labeled 1.1 for you to finish. So for our next large section under cell structure, we need to look at prokaryotic cells. The main difference that we want to remember going into this part of the unit is that they are unicellular organisms. Unicellular organisms means that they're existing as a single cell, whereas most animal and plants that we've just looked at are multicellular, or they're made up of many of those cells that we've just looked at. They're also distinctly smaller, 1 to 5 micrometers in size. You need to make sure you remember that, as you could be asked in an exam question, so you can make a note. Just when we're talking about all of these cell structures, I'm going to disappear quickly so you can see the list. We are not going to call these 4 structures organelles because they don't contain membranes: cell wall, circular DNA, ribosomes at a 70S size, and a plasmid. Now, because none of it contains a membrane, we don't call these organelles, which sounds strange because some of the organelles that we talked about in the eukaryotic organisms didn't have a membrane either, but it's what we are asked to do by Cambridge. As such, let's look at these organelles on the diagram here, and once we've made our way through these, we're going to clarify some of the differences between prokaryotes and eukaryotes for you. So our first organelle, we're going to take a look at those ribosomes, which we already mentioned. Nicely, they carry out exactly the same function, protein synthesis, but that function, whilst it is the same, doesn't occur in the same place. Ribosomes in a prokaryotic cell are not found attached to the RER. Instead, they are free floating within the bacteria. It's not often that you have to recognize a bacterial diagram because they're so small, but if you were trying to look for ribosomes, you would find them in the cytoplasm of the bacteria, not attached to a membrane. Another comparable organelle that we need to make sure we know is the cell wall found in prokaryotes. It carries out the same function in that it provides structural support, but it's important for you to be able to identify it's made of a different material than that in plants. In plants, it was cellulose, and in prokaryotes, it's peptidoglycan. That is most commonly the tested point you'll be asked for. And photomicrograph visual, again, not something you're necessarily going to have to identify, but you've got that large, darker line around the outside of your bacterial cells, which is your cell wall. We also have circular DNA. Now, why are we calling it circular DNA and not just DNA? That's because bacteria do not have a membrane-bound nucleus like the eukaryotic cells do, which is the main defining difference between prokaryote and eukaryote. The function of that DNA remains the same in that it's there for replicating itself, for division of the cell, and it controls the cell's activities. So it's containing all of the information needed for the bacteria to divide, but it's circular, not linear, and it's often referred to as a chromosome. The photomicrograph visual I've given you, you won't be able to distinctly see that there is DNA. However, there is a chromosome within this bacteria, which if you look closely enough can be seen towards the center. Please don't worry too much about any of these prokaryote or bacterial photomicrographs. I've never seen them come up in Cambridge as a question yet, only on diagrams, not on photomicrographs. Now, we've just mentioned the bacterial chromosome, or the DNA, circular DNA, but there is another DNA-based structure found in prokaryotes. It is called the plasmid. The plasmid is a smaller, circular piece of DNA, often just a very, very small loop, And the reason or the function for having that is because bacteria can transfer genes of interest to each other using a plasmid. So it's kind of like bacteria's way of sharing special traits, which are found as genes, between both members of the same species as well as members of different species of bacteria. I did find a really good photomicrograph for you just so you can see it, which is a nice little loop. I'll disappear for a second, just there. However, once again, please don't worry yourself too much about identifying this on a microscope diagram. Now, that was the 4 cell structures we have to know, i.e. those are the structures that you almost always find in bacteria. I have included another one here, which is the flagella, because it does come up in diagrams quite often and is questioned, and that is that it enables motility, or it's for locomotion. It's at the expenditure of ATP, not energy, remember that's what we're trying to say, and it's the way that bacteria move around. You can see a nice diagram just here of a bacteria or prokaryote with a flagella. It's just worthwhile mentioning before I close off this section of the unit that there are a lot of potential structures that bacteria could have that are not necessarily named by Cambridge in their specification. So just be aware when you're doing some more extra research or preparing for exams or revising, you may come across questions that do have some additional organelles. When I say organelles, I should be saying cell structures, but they're not specifically in the specification, so you should almost always be given some contextual knowledge about them within the question. If there's an example that you find that you think I should have gone over, please feel free to leave me a comment. Now that we have closed off this section of the unit, your task is going to be to identify prokaryotes versus eukaryotes based on and as well as identifying the structures. Again, if you are following along with my worksheet, that will be task 1.2. If you haven't got my worksheet, go on the internet, see if you can find some diagrams or some comparisons and have a go yourself. So we've finished reviewing our prokaryotes versus eukaryotes. Now let's take a look at the other structure that we need to know, which is the virus particle. Notice I've said structure. I am not calling it a cell because according to Cambridge, it is not a cell. We'll look at that in a moment. So the components that we need to know for our virus are above me, just up here. We need to know the DNA or RNA structure, the protein capsid, the phospholipid envelope, and surface proteins. There are 4 things written there, but I've only got 3 on my diagram. I will allude to why that is in just a moment. So our first structure, linear DNA or RNA. You may have already picked up on the difference that you have between prokaryotes there. Prokaryotes use circular DNA and eukaryotic organisms do in fact have linear DNA as well. So you can see a pending question from Cambridge there. It's much shorter and smaller because it just contains a few instructions for its own replication, and that replication is carried out by a host cell. So viruses need to infect or enter other cells to be able to be replicated by the host's machinery. That's one of the reasons why we probably don't call a virus its own self or living organism. You don't need to recognize a photomicrograph visual, but just for fun, I have found a few of those for you. So here it is. Our second structure is a protein coat. Protein coat, sometimes called a protein capsid by Cambridge, is that layer around the outside which sometimes plays a role in recognition or binding to the host's membrane, and its job is effectively to protect the material, that genetic material, the DNA or RNA, that is inside of the virus. Again, a nice interesting photomicrograph visual for you there. That pink area in the middle is what we are talking about. Now, we've just mentioned that there's a protein coat, and now I'm going to talk about surface proteins. Really, they're part of the same structure. The surface proteins are a part of that protein coat we've just discussed. However, their function is the specific role of binding to the host cell's membranes and allowing virus particles to insert their genetic material or to enter into that host cell. So they are useful for the virus, But the reason why they get picked up as a separate structure by Cambridge is you want to talk about surface proteins as also as a way that the virus can be identified itself by other organisms or other cells. Particularly, this will become important in the last topic of this unit, which is immunity, because white blood cells could recognize a virus from its surface proteins, which we're going to call slightly differently to the protein coat. Again, a nice little photomicrograph visual for you, shows you there what that looks like with the orange layer around the outside, supposed to be the protein structures or surface proteins we are referring to. Now, those three organelles that we've just finished are the ones that I directed you to at the start of looking at virus particles. However, it's important to mention all of the things that Cambridge have referred to in their specification. As such, we do have one last one here, which is called phospholipid layer. There's a reason why I kind of left it out, but I want to mention it. It is something that does exist in some viruses, and it exists in some viruses when a virus has replicated itself inside of a host cell and leaves the cell. It will take some of the phospholipid layer, otherwise known as the cell membrane, with it when it leaves. So it could exist for many viruses and plays a role in potentially entering into other host cells because it uses the membrane for part of that function. However, I haven't ever really seen it in a Cambridge question before. As such, it's not something that I would spend too long worrying about. However, you've got the breakdown for it, so hopefully now you can add that into your notes if you feel it's necessary. But as I say, it's not commonly tested. So guys, I've made myself really big as I just wanted to address well done, first of all, for getting through most of the bulk of the knowledge that you need to learn for this unit. We only really have to look at microscopes. However, you can see a big exclamation mark behind me. We're just going to go over a few really important points from what we've learned that I know Cambridge question quite commonly to help you as best as you can prepare for that end of year exam when you get to it. These points are an opinion in a lot of examples of things that I think are really important, or clarification points of some of the things that we've mentioned previously which may not have been explained fully just yet. So I'm going to make myself small and drop down to the corner again, but make sure you've got your pens at the ready. All of these are, in my opinion, really important for you to do well in this unit. So I've made myself smaller and then I'm going to disappear straight away. We're going to look at these 5 things for our clarification points. We'll look at them each one at a time, so I'm not going to read them off the slide for you. Now, the first thing on that list was mitochondria and chloroplasts and their evolutionary evidence. What on earth did I mean by that? First of all, let's recap some of the things that we made notes on. Mitochondria and chloroplasts both have circular DNA, which is different to the nuclear DNA that we found in eukaryotic cells. So that's our first giveaway, something's up here. They've got 70S ribosomes and they've got their own ribosomes instead of 80S ribosomes, which we find in eukaryotic cells. You can start to see a trend here. Last but not least, they have their own double membrane, which we believe was formed during entry into eukaryotic cells. Now that leads us to the following conclusion point. These features support a theory called the endosymbiotic theory, which proposes that basically mitochondria and chloroplasts used to be bacteria or prokaryotic cells that evolved to live within eukaryotic organisms. As such, points 1, 2, and 3 therefore support that they are in fact prokaryotic organisms. That can be questioned really commonly in a variety of different ways. If you are going to follow along with my course, I've got some of those questions in my end of unit exam, but please make sure that you're aware they're looking to trip you up here. So our next point was drawing a double membrane. Technically, this is the only skill that I've included in this PowerPoint, but because it's so related to the knowledge of the fact that there are double membranes, I did make the choice to put it into this PowerPoint, not the skills one. You could be questioned on a number of double membranes. Most commonly would be the chloroplasts and mitochondria. I've opted to draw you a mitochondria, so pay attention to the white box on the right. I'm going to disappear so you don't have to look at my face while we do that. While you are watching this video, The most important point is that you do have two separate lines and that they don't touch. The outer layer is in black, or the outer membrane, and then my red inner membrane doesn't touch that one. Next on our list is cell wall composition. You already know it, but I thought I'd add it in here for clarification because it gets questioned so often, guys. You've got cellulose for plant cell walls, peptidoglycan for prokaryotic cell walls. Plant cells and prokaryotes share a feature, but that feature has alternative chemical composition. Guess what Cambridge are going to want to do? Give lots of questions which are going to try to confuse you based on the wording. So if you ever get a cell wall question and you know they could be talking about plants and bacteria or prokaryotes, just take the time to make sure you're happy with what it is they're asking. Last but not least, on our clarifications and preparing for any questions, you've got the nucleus. The nucleus is found in eukaryotic organisms. That is actually what the word eukaryotic means, to have a true nucleus. Prokaryotes do not have a nucleus because there's no membrane around that circular DNA. It's just found within the cytoplasm. Therefore, the DNA of prokaryotes is referred to as circular DNA or a single chromosome, whereas eukaryotes have a nucleus. Make sure that you remember that. It is very commonly tested. So after clarifying the difference between a nucleus-based organism, eukaryotic, and a non-nucleus-based organism, prokaryotic, it's important to remember that there are different names for that genetic material which are found in eukaryotic and prokaryotic organisms, as well as sometimes viruses. Eukaryotic organisms all contain linear DNA, whereas prokaryotic cells will contain circular DNA, and viruses contain a short amount of linear DNA or possibly RNA. It's not ever both, but RNA is a different type of genetic material which we will learn about in a little bit more detail in the course. Exam vocabulary: eukaryotic cells have a nucleus, whereas prokaryotic cells, we call it a circular chromosome. It is sometimes called a nucleoid region by Cambridge. And then last but not least, the virus's genetic material is exactly as it is on the tin. It's linear DNA or RNA. I just disappeared so you could make note of that table if you needed to, guys. The wording in this part of the topic, or naming genetic material, often throws a lot of students off. So just before you move on, make sure that you are truly happy with exactly what we're talking about, as well as how we're naming it as well. And then our last point: why do we not call viruses living organisms? Now, it's very easy to have an opinion on this, and it's good if you have your own opinion about what viruses are, living or non-living. However, we are following a specification, which is Cambridge, and Cambridge deduced that a virus particle is not a living organism. Don't ever call it a cell. They lack organelles, which they need to survive on their own, i.e. they have to use another cell to replicate themselves. As such, you can cite that sentence if you're ever asked, but leave your opinion out of it. Cambridge want you to refer to viruses as non-living organisms. That's brought us to the end of all of the eukaryotes, all of the prokaryotes, as well as all of the virus structures. I would suggest that at this stage in the course, you really want to be able to, from memory, create a big table-based review of all of the structures that you've talked about and where they appear. If you're following on with my worksheet, I've already made that table for you, but I haven't filled it in. This is also a good spot for you to stop and really take a rest, and then come back to the last section of recall information, which will be microscopes after the break. So guys, this is our last section to look at on cell structure, which is on microscopes. When we are looking at microscopes, there are 3 different microscopes which we need to know, and we're going to look at all of them: the light microscope, the transmission electron microscope, and the scanning electron microscope. I do just want to reiterate what I said earlier. This is not going to be the skills for these microscopes. This is simply the information you need to know, and a very quick clarification on what we mean by a couple of terms within microscopy. So, microscopes 101, let's get started. you probably recognize this little microscope from your labs if you have one. This is a light microscope. A light microscope is the most basic and common microscope used to identify specimens. As the title suggests, it uses light. It's very cheap and affordable, and some values that you need to remember are that it magnifies up to 1,500 times, and its resolution is 200 nanometers. This is a common diagram produced, or sorry, a common microscope image produced by a light microscope. It can be colored because you can stain the cells or things that you're trying to look at. Next, you have one of these big looking pieces of equipment. This is a transmission electron microscope. A transmission electron microscope passes electrons through a vacuumed specimen. Because it's vacuumed, it can't be a living specimen, which is a very, very obvious difference between light microscopes and electron microscopes. They're very, very expensive, so expensive that you've probably never seen one. They can magnify up to 500,000 times, and their resolution is 0.1 to 0.5 nanometers, which is very, very small. This, which is very similar to a lot of the diagrams I was showing you on the micrographs earlier, is a common picture that you would see. The pictures produced by an electron microscope are never colored. If they are colored, then it's false imaging. What that means is somebody has come along and colored the picture in. The images are always black and white if taken directly from the microscope. Last but not least, we have our scanning electron microscope. It works in a similar way that it uses electrons to identify the specimen, but instead of passing the electrons through the specimen, it actually bounces the electrons off of the outside. Those electrons are picked up by a large computer, and that then puts together a 3D image. Again, those cells or structures that you're looking at within the microscope must be vacuumed, so they'll be non-living. These pieces of our equipment are very expensive too. Some slightly different values. It can magnify up to 100,000 times, and it is 3 to 10 nanometers of resolution. The images that you would get would look something like this. They're almost always false colored because it's a computer putting together this image. So it's not a real image that you're seeing, it's computer generated. Now, at every single one of those microscopes at the bottom, you saw two phrases which had some values: magnification, which you probably remember from my GCSE, and resolution. Let's make sure we understand exactly what they mean and we can tell the difference so that when we're questioned, we know what to say. First of all, magnification. This is probably something you remember from my GCSE. Magnification is how many times larger an image appears when compared to the actual size of the object. Basically, when you're using a microscope, you're enlarging what you see. So when you say magnifying, it's how much bigger the picture is than in real life. When we talk about magnification in terms of a visual context, this is a picture that when magnified, I have simply zoomed in on. I've intentionally not edited the resolution of this picture. There is no difference in clarity or resolution, we'll look at what that means in a second, between the first and the third image there. Now, what do I exactly mean? That brings us to resolution. Resolution is the ability to distinguish between 2 points that are close together. If you have a higher resolution image, that means you can see more detail. That resolution is usually defined by what you are using as a medium for the magnification of the image. In light microscopes, it's using visible light. In electron microscopes, we use electrons. So, how do we define that in comparison to magnification? Magnification does not give you any more detail unless your resolution is higher. And again, a visual to help you. Resolution here is very low. What do I mean? We can't really tell what any of those parts of the image are. But if we increase resolution, as we're moving from left to right, you can now quite clearly pick out different cell structures and potentially even some different cells. That's because in this, we have increased the clarity, or what we would say in the context of Cambridge is we've increased the resolution. Hopefully those pictures as well as the definitions, which you can make notes on, will make that a little bit clearer. The best way for you to be able to remember this and to learn it is to go ahead and take a look at some pictures of cells from both an electron microscope and a light microscope, and try to talk to yourself about the difference between resolution and magnification. If you are following on with my tasks, that is exactly what I want you to do now. Compare microscope images, and the second part of that task is to use the word magnification and resolution to deduce the difference between which microscope was used. So that should bring us to the end of our cell structure and our basic learning of information points, which are: know the names and the functions of all of the organelles, compare the cell structures across kingdoms, evaluate our viruses as non-living, and then describe the differences between microscopes and know the difference between what magnification and resolution are. Congratulations, guys. I do also need to give credit to Wikimedia, Pixabay, and FreePic, as well as ChatGPT, which is where I got all of my images that you saw from. So I just wanted to take one last opportunity to make my head a little bit bigger again on the screen, and also say thank you for watching this video all the way through. There will be another shorter video for Cell Structure Unit 1 on microscope skills, so those studying Cambridge biology who need to prepare for practical paper 3. I'm going to attempt to teach you how to use a microscope and the skills you need whilst on YouTube, which should be interesting. And aside from that, if you are looking for more helpful biology tips and some other resources, if you search on TikTok or Instagram, you will find my page under Cambridge Biology Revision Mastery, and I also have a couple of places on the internet where if you search for Cambridge Biology Revision Mastery, you can download all of my PowerPoints and slides. Thanks very much, guys. See you next time.